Welcome to today's Cell Culture series by MusB Research. In this video, we're going to break down one of the most important concepts in cell culture, the difference between adherent and suspension cells. If you plan to work in biology, biotechnology, microbiology, or biomedical research, understanding these two systems is essential. Why? Because the way cells grow determines how you design experiments, how you handle them in the lab, and how you interpret your results. Let's begin. What are adherent cells? Adherent cells, also called Anchorage-dependent cells, must attach to a solid surface to survive and grow. In the lab, they attach to specially treated plastic surfaces like tissue culture flasks, multi-well plates, culture dishes. These surfaces are chemically treated to allow cells to stick. Most cells that come from solid tissues are adherent. For example, epithelial cells, fibroblasts, endothelial cells, many cancer cell lines. Key characteristics of adherent cells. Here are the key features. 1. They grow as a monolayer, meaning one flat layer across the surface. 2. Their shape matters. Changes in morphology can signal stress, differentiation, or transformation. 3. They must be detached using enzymes like trypsin or acutase when passaging. 4. Their growth is limited by surface area, not liquid volume. Once the plate is full, they stop growing. This is called confluency. Next, trypsinization. When adherent cells reach about 70 to 90% confluency, they need to be split. This process is called passaging. Here's how it works. 1. Remove old media. 2. Wash with PBS to remove serum. 3. Add trypsin, an enzyme that breaks cell attachment proteins. 4. Cells round up and detach. 5. Neutralize trypsin with fresh media. 6. Split into new flasks. This step is unique to adherent cells. Suspension cells don't need this. What are suspension cells? Now let's contrast that with suspension cells. Suspension cells, also called Anchorage-independent cells, float freely in the liquid culture medium. They do not require attachment to survive or divide. Most blood-derived cells are naturally suspension cells, including lymphocytes, hematopoietic stem cells, leukemia cell lines. Key features include 1. They grow as single cells or clusters in liquid. 2. They are passaged simply by diluting into fresh medium. 3. Growth is limited by nutrient availability and volume. 4. Cell density is extremely important. If the density is too low, cells may not survive. If too high, nutrients run out and waste builds up. Let's compare them directly. Adherent cells need a surface to attach to in order to grow. They typically spread out and form a single layer, called a monolayer, on the culture dish or flask. Because they stick to the surface, they usually require enzymatic treatment, such as trypsin, to detach them during passaging. Their growth is also limited by the available surface area, which means scaling up can require more culture vessels. Suspension cells, on the other hand, grow freely floating in the culture medium. They do not need attachment, and they are usually passaged simply by diluting the culture into fresh media. Their growth depends more on volume than surface area, which makes them easier to scale for larger production systems. This is one reason why suspension cultures are often preferred in biotechnology, especially for large-scale protein production and industrial applications. Understanding the difference between these two growth types is essential for choosing the right culture method for your experiment. Experimental implications. The culture system you choose directly affects your experimental design. For example, transfection efficiency can vary between adherent and suspension cells. Drug exposure may change depending on how cells interact with surfaces or with each other. Imaging experiments are usually easier with adherent cells because they remain fixed in one place. And for large-scale biologics manufacturing, suspension cultures are often used in bioreactors. So ultimately, the biological question you're asking should determine which culture system you use. Important biological concept, Anchorage independence. There's an important biological concept to understand here. Normal epithelial cells usually require attachment to a surface in order to survive and grow. But some cancer cells acquire the ability to grow without attachment. When cells can proliferate independently of a surface, they often show increased tumor-forming potential. This property is known as Anchorage independence. Scientists commonly study this using a soft agar colony formation assay, which allows researchers to observe whether cells can grow in a semi-solid environment without attachment. To summarize, adherent cells depend on surface attachment to grow. They typically form monolayers and require enzymatic detachment during passaging. Suspension cells, in contrast, grow freely in liquid culture. They are usually maintained by simple dilution and are easier to scale because their growth depends on volume rather than surface area. So choosing the right culture system ultimately depends on your biological question, your experimental design, and your scalability needs. Understanding the difference between adherent and suspension cells is foundational for cell culture. In our next MusB Research video, we'll cover aseptic technique and how to prevent contamination in cell culture. If you found this helpful, subscribe to the MusB Research Cell Culture series. Please like and share it with others who might benefit from it. We'd also love to hear your thoughts, so feel free to leave a comment below. Thank you for watching, and we'll see you in the next episode.